I'm going to get us started here. My name is Jenna Crowder. I am an apiary inspector with the Utah Department of Agriculture and Food and the Western Regional Director of the Apiary Inspectors of America. And I also run our in-house diagnostic lab at the Utah Department of Agriculture and Food, where we offer diagnostic services for beekeepers, both in-state and out-of-state. I'm also going to be joined today by James Fulton from Florida. James, if you want to give yourself a little intro while I get my screen sharing set up here.
Hi, everyone. I'm James Fulton. I am the research scientist with the Florida Department of Agriculture Honeybee Diagnostics Lab. So we do diagnostics for diseases and pests that affect honeybees. We also do some testing in bumblebee species, and we're involved with regulatory as well as non-regulatory concerns in and outside of the state. So I'm just here to speak a little bit about one technology that we use.
Sweet. Thank you so much for being here and for your time, James. So we're going to I'll start with a little bit of an intro. Today we're going to talk about there we go. About Tropaelaelaps monitoring methods, diagnosis of the mite itself, so what to look for, what the mite actually looks like. And we're going to touch on management a little bit. All right, so as a little recap from last week's webinar, just to refresh in your minds why Tropaelaelaps mite is a pest of concern. Its current distribution is in the Asiatic region and in Indonesia, but it is expanding its geographic range, which is worrying. And it is also problematic in its native range, causing problems for beekeepers in those areas where it is extant. It reproduces faster than Varroa. It has a quicker reproductive cycle, which means that it could experience rapid population growth once it's established in a colony. Another challenge about Tropilaelaps is that, similar to Varroa, it feeds primarily on brood underneath the wax capping, but it lacks a phoretic phase, unlike Varroa might. So it's not spending time hanging out on the adult bees. It's spending the majority of its time underneath those cappings, making it very, more difficult to detect using traditional detection methods like we're used to with Varroa mite in the United States. It also, moves very quickly on the frames. So even while it is outside of the wax cappings, it can be difficult to detect just by its rapid movement, especially when you're looking at a frame that has bees and resources and a whole bunch of other stuff on it. It can be difficult to detect these very, very small mites. And of course, the main reason why it's of concern is that it does pose significant impact to the colony health. It can cause damage that looks very similar to Varroa type damage. This is called a Varroatropy complex. So kind of similar to that parasitic mite syndrome look, slumped larva, signs of parasitism, spotty brood pattern. And similar to Varroa, it is known to spread viruses and has been found to be in association with deformed wing virus, acute bee paralysis virus, and others. It can kill developing pupa while they're under the capping, and simultaneously, it can inflict feeding wounds on the pupa, even if they do make it to adulthood. So you can end up with bees that are stunted in growth. They have wounds on antenna, legs, appendages, the body, and through those, in those feeding wounds, the viruses can be vectored as well. So you can get adult bees with deformities, which is kind of unique to Tropy. All of this combined can result in rapid colony collapse, and unmanaged infestations can quickly kill colonies. Thankfully, this is not in the United States currently, but it is something that the regulatory apiary world has kept a very close eye on recently, and we want to be proactive about spreading awareness of it. Since there are, of course, more beekeepers than there are regulators who are looking at hives. So we're trusting our extension agents to also spread this knowledge and communicate the word about Tropilaelaps mite and communicate to our beekeepers. Here's some images of Tropy mite on a sticky board. This is one of the more effective ways of detecting the mite, keeping in mind that it's difficult to see while it's alive on the frames. It's difficult to see when it's alive under the cappings, unless you're doing brood uncapping, which is very time consuming. But even sticky board monitoring can be very time consuming and difficult. The mites themselves are very, very small. We've spent quite a good deal of time in the various workshops inspecting sticky boards, and I'm going to have us look at some sticky board photos and real sticky boards today, and you'll experience firsthand just how difficult it is to detect these mites. I swear I get them confused with just pieces of pollen or debris from the hive whenever I'm looking at these boards. So it really is difficult to detect these mites using a sticky board, even though that is one of the more effective ways to detect them compared to something like a sugar shake or an alcohol wash. The bump test is another monitoring method that is effective for detecting these, but it can cause damage to the colony by killing larvae. So this is something that for folks who have done the National Honeybee Survey, this is part of that protocol, and you may be familiar with it there. It involves really slamming a frame of brood against a pan of water and knocking the larva out. So you have to inherently damage the brood to get a good sample. It can also damage the comb. This can not be great for the beekeeper's perspective, which is totally understandable. And then it's also time consuming to go through these samples in the lab under a microscope and look for the Tropilaelaps mites, even once they're filtered through a cheesecloth, and we can pick through those larvae to look for the mites. It's still pretty time consuming. And it is also going to be less effective because keep in mind those mites are going to be running around on the frame or underneath the capping. So you have to really smack that frame to get a good sample. Other forms of monitoring that were also discussed briefly in the previous session would be eDNA. So this is arguably the most effective detection method, but it is costly and few labs offer the service. And we're going to have James talk a little bit more about that in just a bit here. Uncapping pupa and brood comb is also effective, but that is very time consuming and it can be difficult to see the mites in the field. Even under the microscope, it's difficult. Sticky board is nice because it's more of a passive monitoring method, but it is, again, effective and time consuming and, once again, difficult to see these mites. Visual inspection of the frames themselves, unless you have a really trained eye, it's going to be difficult to see those mites. And when bees are on the frames, it is nigh impossible to see these mites underneath the bees themselves. The bump test, slightly less effective, and it can damage the comb and the larva. And then sugar shake and alcohol wash, you can get some tropy mites. There's been a study done by Auburn University comparing some of these monitoring methods, and they found that, yes, you can find tropy mites doing a sugar shake or alcohol wash, but compared to all of the other methods, it is the least effective because, again, these mites lack that phoretic phase, so they're spending very little time on the adult bees. So now what with this information? If monitoring is very difficult and it's hard to detect these mites and we have a limited selection of monitoring techniques, we start to think about management, right? Again, with the short phoretic phase and how much time these mites spend under the cappings, that presents unique challenges for management. The most promising options are formic acid, potentially combined with oxalic acid treatments. But again, that short phoretic phase limits the effectiveness of treatments that target mites while they're on bees. I'm thinking things like thymol. I'm thinking things like Viroxan, the recently approved oxalic acid extended release. These things that work on the phoretic phase, they're not going to be as effective for this mite. And then just kind of speaking from personal experience, formic acid can also be challenging because it has temperature restrictions with it, right? I know in Utah, it's very difficult to find a perfect window of time in mite season when it's below 85 degrees and you can safely use formic for a week or 2 weeks. So these are things that we need to keep in mind when making management recommendations. There are also integrated pest management strategies that are utilized in the tropy's native range, such as keeping small colonies and doing frequent brood breaks, but those strategies likely would not translate well to commercial U.S. beekeeping, which is another challenge with tropy. The long and short of it is that more research is needed for monitoring methods to control this mite. If you suspect you found a tropy mite, so let's say that a beekeeper has reached out to you as an extension agent or something like that and thinks they found a tropy mite, the key is that in order to officially get USDA confirmation, so federal level confirmation that the tropy mite is extant, and then in return or subsequently federal level help to deal with it, whatever whatever that means for the state that it's found in, a physical specimen is going to be required. So the first step would be collect that specimen. If someone thinks they found a trophy mite, you can use all of these other monitoring methods that we discussed, like doing a brood bump, collecting hive debris, uncapping brood, and collecting those pupa. And really, the more sample types that you can collect, the better. Again, keeping in mind that it is going to be not one of these methods is going to be the perfect method. So better oversample and maybe actually find the specimen than undersample. Storing the specimen is going to be important so that it safely makes it to its official lab, wherever the USDA says, hey, send it off to this lab for official confirmation. So that could be keeping it dry if you're dealing with hive debris, but ideally keeping it in a container with alcohol, or putting it in a freezer if you're going to be storing it dry. And then, of course, report it to your regulatory official, your state apiary inspector or apiarist, and they will get the process started on getting that official confirmation, communicating with USDA, and starting to work on a mitigation or management strategy. And really, that is going to be the refrain over and over again, is always reach out to your state apiary inspector, because the immediate response will likely be different in each state depending on where this thing is found first. Now I'm going to turn it over to James Fulton from Florida to talk a little bit about eDNA sampling. As we said, this is one of the more effective methods, but it does have some benefits and drawbacks. James, I am still in control of slides here, so whenever you need me to advance, please say the word and I'll click forward to our next slide.
Thank you, Jenna. Yeah, so we can talk about eDNA. Would you advance to the next slide? So we are currently using eDNA methods for two different things. We have a Farm Bill supported project looking at swarm traps that we've deposited throughout the state, and we're using it as a sentinel detection program, trying to see what pathogens or pests might be present. And we were also testing it with the University of Florida to see if it would be an indication for Vespa velutina hornets that were hovering over hives or nearby. So I was asked to be involved with this aspect, or at least describing it a little bit, since I have personal experience. And given the limitations of how we're trying to identify Tropaelaps using the brood bump or hive debris, we are currently looking for improved methodologies that can make things easier or more efficient. And so one of these ideas is environmental DNA. And that's a term that's bandied around quite a bit. And so a brief explanation of what environmental DNA is whenever an organism passes through an environment. For example, if a human is sitting down at a desk, we put our hands or arms on the table, and our skin cells will be deposited, and that could include the intracellular components of our skin cells, mitochondria, nuclei, ribosomes, all the way up to our pathogens as well. And so the whole idea has been, can we use environmentally deposited DNA that has been deposited by organisms passing through an environment as a way to detect them when their physical presence, their physical entity themselves is not available? So for example, with pathology testing, we have typically taken an organism, the host, and extracted DNA from that individual for pathogens of interest. And an eDNA approach would be to swab the external surface of that honeybee and to test it for pathogens. So we may not actually need the physical intact specimen present to do the testing, but we could take swab samples. So Jenna, if you would move to the next slide. So environmental DNA methodology has really been have come of age through the conservation biology movement. It's been used particularly for the identification and detection of cryptic species, or those that are endangered or in very low numbers, because the amount of material that you need is very limited, and it might be difficult to find that endangered species, with a pair of binoculars or on video recording. But if you're able to record or to take a sample of DNA and then test it through something like sequencing, you might be able to confirm that that individual has been present, even if you haven't physically seen it. And so a lot of the early work started as this is a conservation tool, and it's now become something that we've used for testing of pathogens in the air, in water sources. So looking at, for example, with COVID, when we went through our COVID epidemic, they were testing water samples from sewage systems, and this is a form of environmental testing. So we're not testing the patient themselves for COVID. We're testing, water or sewage that might have the deposit of viral proteins or nucleic acids. And so now we're seeing this technology emerge. And our question is, can we use this for Tropaelaps detection? So if we move forward, Jenna. So in the apiary yard, there are different surfaces or substrates that can be used for collecting environmentally deposited DNA. And like I mentioned, that we're doing it on swarm traps. I should have put a photo on this presentation, but I didn't. We are swabbing the outside of swarm traps. And I have a picture here in a moment of what those swabs look like. But we're just swabbing the outsides of those swarm traps, and the entrances of the swarm traps, and then we're going to compare it to whole bees, and we're going to see to what extent are we matching results. But if we look at an apiary yard, there are other surfaces that could be promising, and that could be the honeybee hive entrances. It could be the sides of frames. It could be the baseboards. It could be hive tools. Hive tools are, we've even seen this with Vespa velutina in Georgia. We're able to detect to a limited extent the hornet DNA on hive tools. Hive tools get everywhere, and they really do magnify and seem to pick up everything that's in that apiary. You could also consider water sources. So, every organism needs water, and so a lot of times water sources can be a great conduit or accumulator of material, like a bioaccumulator. So you could test that. You could also test nearby foraging material. These are all possible areas to look for in an environmental DNA project. And this just to kind of bring it to you in the context of an apiary yard. So Jen, if you can move forward one slide. There are positives and negatives with this methodology. So as Jenna mentioned, official identification is only going to occur with the hand specimen intact at a USDA APHIS facility. So even if you had nucleic acids that sequenced as Tropaelaps, that would not be considered a positive identification. You would need that physical specimen intact for that identification. Other concerns, other drawbacks could be that laboratories that are doing this kind of work need specialized equipment and personnel. They need to be comfortable with DNA extraction, with basically hygienic techniques. They need to be able to use molecular assays like qPCR machines or Illumina sequencing technologies. And that kind of takes us to the next point, which is that this methodology can be expensive. And also the concern that the limited persistence of DNA. So DNA or RNA doesn't last or survive very long in the environment. However, this drawback is becoming less and less of a concern. So we're developing techniques that can extract DNA from surfaces for greater and greater periods of time, but it is still something to keep in mind. So positives is that the methodology can be much faster. So with brood bumps, it can take 15 minutes, first calming down the beekeeper that you're not going to kill other bees. But then you have to open the hive, and you have to take out frames. You have to find a frame that has brood. Then you have to, forcefully dislodge that brood debris into a container to collect it. It can be a pretty time-consuming process, as well as looking at bottom boards. Whereas with the eDNA approach, it could be a matter of just taking some very simple, quick, efficient swabs of a hive. speeding up the whole process quite a bit. It also doesn't require somebody to have experience identifying, based on morphology, Tropilaelaps. All they would need to be able to do is take a swab, and then they could send it into a lab, and then that lab could process that material. So they wouldn't need to have the comfort or the expertise to be able to identify it. It might be a greater sensitivity. So one of the drawbacks with the brood bump is that it's not capturing to 100% the presence of all Tropaelaps, which means that there is a risk that a particular beehive or colony is infested, and we just don't detect it with that method. With eDNA, this is being tested, and evaluated and validated. But the idea is that it could be more sensitive than that methodology. And that would allow for things like early detection. So if we could detect it in a single apiary, just starting off as an infestation, it might give us greater time to launch an eradication effort. Whereas if we wait for the physical presence to be visible, at that point, the infestation levels are pretty high. So yeah, there's drawbacks, but it definitely has some considerations here. Jenna, if you could move forward. So it's pretty simple to collect a sample. Um, there are different types of swabbing swabs that you could get. So in the picture on the left, there's just different companies that make these forensic swabs. I'm not going to endorse any particular product, but there are different companies that you can purchase this material. And the way that we've been doing it is that you really essentially only need molecular water, because you don't want to degrade your DNA or RNA. And that water also moistens the filters that the swab is on. It moistens it so that it can better absorb, like a sponge, any material that might be present. Gloves, your swabs, labeling, so a pen or pencil, and then some kind of collection device to keep all your stuff together. So it's you don't need much to collect a sample in this way. And so I went out and I took some quick pictures on Monday, actually, of doing this process. So the top left is you're just starting with your swab, and the blue 50 mil Eppendorf tube is molecular water. And so in the second picture on the top, I'm just showing that I'm moistening that swab with that water. You can moisten it for 60 seconds. And then in the third photo, in this case, I was swabbing the hive entrance where honeybees would be coming in and out, where mites may or may not be leaving or entering as well. And then I have a series of photos here of other locations that could be swabbed, including the sides of frames. That I have at the bottom left is the front image of a frame with brood. And so I'm just swabbing the cell coatings, or the cappings of the cells, where the honeybees are congregating, where the brood is, and then also taking swabs of the bottom board. And so these are all locations that swabs could be taken from. And it really is that simple. You just you could take several different swabs and run the swab over these areas for 1 to 2 minutes until the swab is discolored to a bit, so you can see that it's picking up sediment. And then you would put it back into its container, and then put it into a Ziploc bag. Ideally, you would keep this in the freezer and overnight it. But yeah, I mean, 24 hours, 48-hour mail shipment would be ideal. You wouldn't want to keep this on your dashboard in your truck or something, but keeping it in a cooler with ice, and just keeping it environmentally controlled would be the way to go. Jenna, if you could go one more. And then the way that it works is a swab sample comes in, and then we do extraction, DNA extraction in different ways. So we use biotinylated probes, which are magnets, and it binds to the strong phosphate group, and it pulls it out of suspension. But you can do other types of kits like QIAGEN, blood and tissue extraction kits, CTAB, many different ways to do extractions. And then you can test it in various ways. So these are just pictures of equipment that we have in our lab. So we have access to qPCR. You can do basic PCR and gel visualization, or you could do some sequencing. on something like an Illumina machine or a nanopore sequencing. And then you would have that data that would give you the presence or absence information. If it did come back as a hit, if you had a qPCR assay that came back positive, or you had reads from an Illumina sequencing run, what you would then want to do is follow up with a more in-depth visual examination of that apiary, because again, you will need a physical intact specimen for that official confirmation by APHIS. So this data by itself would not give you the information that you need. It is just an early detection method so that you can, follow up with more in-depth resources for that hands-on in-person study. There's different labs that are doing it. So our lab is doing it. Florida Department of Agriculture, the National Agricultural Genotyping Center in Fargo, North Dakota is also doing it. I believe Auburn with Jeff Williams, Dr. Williams' lab is evaluating, validating this methodology. But if you'd like more information, the AIA, Apiary Inspectors of America website does have a list of laboratories that have these capabilities available for you.
Great. Thank you so much, James, for giving us such a detailed look through eDNA. And I think what James said at the very end there of this is a tool that can be used to identify where further investigation is warranted. I think that's where it really comes into play. And with the idea that we really want to detect this thing early. Once we see signs of infestation or we see tropy running around on the frame, it's probably way too far gone. We're talking... Once we see them, it's probably very late in infestation, kind of similar to Varroa, right? So these eDNA tools give us an opportunity to early detect and respond quickly and potentially keep things contained or mitigated. It's always going to be situationally dependent, right? And not every state is going to have the funding to pay for this testing because it is also pretty expensive. But, it's always worth reaching out to your apiary program in wherever state you are located in, seeing if this is something they would consider. I know we are considering it here in Utah. Obviously, Florida's considering it, and it could be a really great early diagnostic tool to detect where we need to do more thorough inspections to find the specimens. Right. Okay. So once you've done the bump test and you've knocked the Tropaelaps mites or hive debris and brood out of the frame, that water gets filtered through a cheesecloth that's in a strainer, so you end up with this mass or this kind of goop of larvae and mites, and then this can be sorted through under the microscope. So this would be a really promising way to get a specimen if you suspect that there are Tropaelaps mites in a hive, potentially indicated by an eDNA positive. Another method could be sticky board monitoring, which I've mentioned a couple times already. The trick with sticky board monitoring is that, of course, you're going to get a lot of debris, and you kind of want to minimize the amount of debris that you're going to get so that it's easier to process this in a lab and actually see the mites because they are, again, so difficult to see on these sticky boards. You want to limit the amount of time that they are installed. So they should be installed only, up to 72 hours max if it's a smaller colony. If it's a larger colony, you want to keep it installed for a less amount of time, again, to reduce the amount of debris that you have to sort through. Here is kind of I wanted to give us a little interactive looking at this section of a sticky board. And this is a good example of some of the fantastic photos that are available on the Project Apis M website in relation to tropy. So this is a very, very zoomed in photo. I don't know exactly the magnification here, but let's see. Can folks see my mouse pointer?
Yes.
Okay, so here I am circling the Varroa for scale. So we do have a Varroa on this frame. So I'm going to pause it here and just give folks a second to kind of scan this visually and try to identify where the Tropaelaps mites are on here. I promise there are some. It looks like there's also some, like maybe some wax moth casings there, some wax moth exuvia. Along with some debris. And then here we can see circled where those Tropaelaps mites are. So we've got one in the top, one kind of in the middle here, one on its ventral side where we can see the dorsum, and then here we've got another one on its dorsal side, and actually two in here, and then a third one, or I'm sorry, a 1, 2, 3, 4, 5, sixth one all the way down here. And you know there might be more on here. Again, I am not the best at detecting these myself. I get them confused with debris all the time. Here's another little test here. So I'll pause it and let folks kind of visually scan this and see if they can pick out where the Tropaelaps mites are. Remembering from the last session that what we're looking for is a mite that's longer than it is broad. They are an arachnid, so they do have 8 legs, but those first 2 legs are held up like antenna. So they almost look more like antenna than front legs. And here we can see where those Tropaelaps mites actually are. One in the top right corner here, one on its dorsal side where we can see the underside, one on its ventral side where we can see the backside, and then another one flipped over where you can see its tummy. The ones where we can see the ventral side are, in this particular image, are great examples of one of the key diagnostic characters of Tropaelaps mites, which is they have this very visually distinct, what's called an anal plate. This is what can be used as a diagnostic character when you're comparing them to some of the lookalikes. So in those ones that we see here that are flipped upside down, they're laying on their backs and we can see their stomachs. That dark line in the middle there, right there and right there, and is the anal plate, which is pretty visually distinct. Some of the common lookalikes that we can expect to see, the most common one is probably going to be a male varroa mite. I have had a beekeeper reach out suspecting tropy, but it turned out to just be a male varroa mite. These are pretty ephemeral, so if a beekeeper sees this, I think it's like kind of a lucky day, like, oh, pretty rare, you found a male. But they are going to be a very different shape. They're going to be much more round. Their legs are splayed out a little bit differently, and they lack that distinct anal plate. Some other lookalikes would be a pollen mite. There have been some case studies recently of beekeepers reporting pollen mites as potential Tropaelaps mites, and then those specimens turned out to be, thankfully, pollen mites, and you can see they also lack that distinct anal plate. Even here, so it is a little bit of a blurry image, but if you can see my mouse cursor kind of highlighting, we can barely see the outline of that anal plate, and I think it is so distinct on the Tropaelaps mites compared to the lookalikes. So that is a really important key diagnostic character to look out for when you're looking at these beasts under the microscope. Okay, now I'm going to do another little screen sharing shuffle because I wanted to take some time to look at actual sticky board pieces and actual lookalike specimens. Again, huge thank you to James Fulton from Florida for coordinating with the insect collection that he has access to. And sending me some slide-mounted specimens of lookalike species for us to actually look at. I also wanted to shout out Dino Light Scopes, not endorsing them in any way. Can't do that since I'm a government employee here, but this is literally the setup that I'm working with right now. And I love these handheld USB scopes in particular because they are very robust. You can take them into the field, and for outreach things like this, they're very easy to plug into a computer. And we'll play a little bit around with this one and take a look at some specimens under here. It's a great budget option as well in that they are relatively cheap. There's a wide range of these types of scopes that can go up to just under like $1,500, but the cheaper ones will range. The one that I'm using today is a sub $600 scope, and I think they are a great option for examining sticky boards and things like this. Okay, so you saw the setup that I'm working with, and this is exactly what it looks like. So I'm sitting over here I've got a section of a sticky board that was graciously sent to me by Rogan Dan Orrell, over at Auburn University. And we've got this at about 30 times magnification here, if the calibration is correct. And we can see that the visual image is pretty good. We can see there is a Varroa mite on here, and we have a Tropaelaelaps mite right next to it. With the sticky boards having this kind of sticky substance on it, the thing I really like about these scopes is that they also have a polarizer that gets rid of that visual noise and makes it a lot easier to see. We can also see here again that anal plate, just the faint outline here, which to me is a very easily identifiable key diagnostic character and would be a reason to send this specimen off to a USDA official identifier, for example. If we compare that to a lookalike, so here we've got bear with me while I get this in focus. Here we have a pollen mite that is very different in shape. So first of all, we can see that so what I've done is brought it into focus, but I have not changed the magnification at all. So it is pretty similar in size, but the legs are splayed out a little bit differently. It looks like it may have a little bit of that anal plate here, but the shape is quite different. This is a specimen that is more round and less of that longer than it is broad body form of the Tropaelaelaps mite. This is a specimen in the family Chilodactylidae. And looks like this one was collected in 1968. So this is quite an old specimen, collected off of a carpenter bee. Here we've got a unlabeled slide here. I'm not sure if James is still on. But this is kind of a cool exercise in what is this thing? Could this potentially be a trophy mite? Kind of a mystery slide here. So bringing this into focus, we can see that the size is about the same. We've got a slightly longer than it is broad, but I am not seeing that distinct anal plate that is characteristic of Tropaelaelaps mite. So this could be one that could be sent off for official identification if you because it's always good to know what it is. But again, lacking that distinct anal plate, this one I would identify as a pollen mite, not a trophy mite. Comparing that to we've got one more specimen I want us to take a look at over here. Where did she go? There we go, right here. So this is another trophy mite that's on its dorsal surface. So we're looking at its tummy here. And we can see again the outline of that distinct anal plate, the way the legs kind of curl in when they are dead on their backs like that. And if we kind of play with the polarization on this scope, then we can see a little bit more clearly the outline of that distinct anal plate. And once again, just for scale, here is a Varroa mite under this same magnification. So now that I've gotten my dino light all sticky here, putting it on the sticky board. Just gonna bring it back so we can take one more look at the two right next to each other, because I think this is a very useful comparison image just to get an idea of the body shape. And then I will go back to my presentation here. Are there any questions, or did folks want to take another look at the lookalikes before I switch back to the presentation? We are almost done here, just a couple more slides to go. So we've taken a look at some specimens under the scope, some lookalikes. some tropy and some varroa. Let's talk about management a little bit. So, primarily the research on this has been around formic acid because it does target the mites, varroa mites, while they're underneath the cappings. So this makes it a promising treatment for a mite that spends most of its time underneath the cappings. However, again, recognizing that formic has its limitations, it can't be used in hot weather. That's the main drawback, right? At least in my experience. This graph here is from a study that Rogan Tokash and Dan Arell and the rest of them over at the Auburn Bee Lab did on looking at different combinations of cultural control methods like a brood break in combination with formic acid and a brood break in combination with oxalic acid. And they found that the most effective options for control were a brood break with formic and a brood break with oxalic. They were pretty equitable or pretty similar in results, right? So the idea that formic is going to be targeting them underneath the cappings, and then oxalic can clean up any mites that might be running around on the frames in that very short period of time when they are outside of the cappings. Again, this is something that is not going to, with frequent brood breaks, is likely will not translate very well to commercial beekeeping industry. So I really think more research is needed on what other management options there are chemically, culturally, perhaps in looking at hygienic behavior. We know that bees can have hygienic behavior for varroa mites, so what about tropy? This is a very new area of research, so there's not a whole lot out there. But right now, the promising stuff is formic and oxalic in combination with brood breaks. Now I'm going to point you, just wrap up here, point you back to some additional resources. These have also been mentioned in the previous presentation, but just to shout them out again, the website honeybeepest.org/tropy is a great, reliable resource for more Tropaelaelaps information, distribution, and the ongoing research on it. So definitely check that out. There are some really fantastic images from in the field, images of more of these sticky boards, and more information about the distribution. And then, of course, shouting out the Apiary Inspectors of America website. When we are dealing with invasive insects like this of regulatory importance, it is so important to reach out and maintain a good relationship with your apiary inspector, to ensure that we can get that early detection, rapid response, and have that government support to on a state level, and then eventually, hopefully, on a federal level, if we do find a specimen and get that identified and confirmed by USDA. So all of that regulatory stuff is going to have to go through your state apiary inspector, and you can find the contact info for your apiary inspector at our website, apiaryinspectors.org. There's that QR code should take you there. And then also, if you just go to that website, apiaryinspectors.org, I believe the first thing on there is find your inspector. So definitely encourage you to check that out. Thank you so much for your time and attention today. I want to shout out Auburn University for all the great work that they've done on Tropaelaelaps research. Chiang Mai University in Thailand, they're also actively researching this, and they're a partner of Auburn University. And then Project Apis M for being a great resource and providing creating and providing these fantastic images that we can reference when comparing, is this a trophy night? What do we need to look for? With a pest that is kind of, I would say this could potentially be a cryptic species, right? It's very difficult to detect. Having these high resolution images is so important. And then, of course, got to shout out Apiary Inspectors of America for doing the outreach work and working to spread awareness about this mite before it hits our shores.